Interstitial chemotherapy allows immediately after surgery, or actually during surgery, to start chemotherapy on what hopefully is a very small residual amount of tumor. If we can get 98% of the tumor out with surgery, and increasingly that's the target of our surgical approaches, if we can do that safely, then it immediately starts working on the remainder, and then it's followed by radiation therapy as well as Temodar systemic chemotherapy, which is relatively well tolerated. Now, going forward, the question is, should the Temodar be given in polymers as well? Should we give that locally? And we might reduce the 2% or so patients who have very serious side effects from Temodar. We might be able to eliminate that. And we might also, and we certainly see that in the laboratory, we might get more effective treatment of that drug if it was given in log order higher concentrations and over a sustained period of time in the brain itself, during radiation therapy as well as afterwards. So I think there's hope with that. Some of the other things that we're looking at is a variety of new drugs that have not been shown to work before. For example, Taxol is a standard drug used for breast cancer, lung cancer, other cancers. It works in those cancers. It works beautifully in the laboratory against brain tumors. Every clinical trial has shown it to be a failure because it doesn't cross the blood-brain barrier. You can't get it in. Good drug, FDA approved, available readily, Taxol or Taxotere, but you can't use it for brain tumor patients, adult or pediatric. We have shown that with polymers, and we've gone into clinical trials with that in a phase 1 trials, that with polymers you can deliver Taxol directly to the brain tumor, and therefore you could take advantage of an existing drug, very powerful, that has no use clinically for brain tumor patients, but may be very powerful to add either singly or in conjunction with other therapies. That would be one example. The platinum drugs would be another example. Newer drugs and newer, glycolytic things that affect glucose metabolism may be very powerful when given locally and to sort of starve the tumor. We've looked at inhibitors of blood vessel proliferation, anti-angiogenesis. How do you use them both locally and systemically? All these are areas of study. The most exciting area that I think right now is work with the immunotherapy and what the role of local chemotherapy is in combination with immunotherapy. So one of the most exciting areas now in oncology is harnessing the power of the immune system using checkpoint inhibitors. And they allow the immune system to recognize the cancer cell and to destroy it. Some of the checkpoint inhibitors like PD-1 have been approved for melanoma, for small cell carcinomas and other cancers, and they're just beginning to be tried for brain tumors in clinical trials. Now, we looked in the laboratory, and this is Michael Lim and colleagues at Hopkins and Drew Pardell, and showed that, in fact, if you give the PD-1, which is a checkpoint inhibitor that's now FDA approved, to animals, you see a benefit, and you see a memory, immunologic memory. If you re-challenge the survivors 3 months later, they'll reject the tumor. If you treat those animals with standard chemotherapy, either carmustine or Temodar systemically, you don't see much of an improvement, and you lose that immunologic memory. On the other hand, if you use a local chemotherapy, either Gliadel, which is the carmustine, or a polymer with Temodar, and the checkpoint inhibitor, there's a synergistic benefit initially. More of the animals survive the initial tumor and reject it, and 6 months later, they still have that immunologic memory. So that puts into question our whole paradigm of how we're treating. If we're going to use immunotherapy, we may not want to use systemic chemotherapy that wipes out the immune system. And in fact, to preserve the immune system but still get the benefits of chemotherapy, we may very well want to combine local chemotherapy with checkpoint inhibitors and see if that combination is more effective at both wiping out the tumor and sustaining an immunologic memory. Now, this is a hypothesis. It's based on animal work, experimental work, but it is moving into a clinical trial now, and we'll have an answer if that is correct or not. We're opening a clinical trial at Johns Hopkins combining local chemotherapy with Gliadel with the checkpoint inhibitors. And that's just going through the regulatory process now of getting approval for the trial, but we have funding for the trial and approval from the hospital for the trial. So I think that'll open relatively soon. And then it takes about 2 years to do a trial like that and to analyze the data, and we'll know. And if it is correct, it's going to make us rethink how we're giving chemotherapy in combination with immunotherapy. If it doesn't work, then we go back to the drawing board, and that's okay too, because the only way we're going to make progress is by trying new ideas. Well, we should be open-minded about whether it should be expanded even initially to children as well. I think that maybe that'll be one of the outcomes of this workshop, is that we have that discussion. Well, I think that the basic biology of the tumors is the same. I do think that the context of the developing brain needs to be considered, and the vulnerability of a child's brain. On the other hand, when you're dealing with fatal diseases, the first objective is to destroy the tumor. If we can't do that effectively, then nothing else really matters anyway, because the child, unfortunately, will succumb to that tumor. So if we have methods of more effectively treating the tumor, we would be optimistic that that would lead to a better outcome. The other thing is, these approaches, such as the immunotherapy and the more localized chemotherapy, make sense that they should be beneficial to children. There's nothing in them that should be more toxic or more worrisome for children, but that needs to be tested. Well, I think that the take-home message is that we've made progress, and it's real progress. We've seen, a significant improvement in the median survival for the most untreatable malignant brain tumors, and we've done it by an approach that goes from the laboratory to carefully designed clinical trials. We're at such a different point in time now that we have a huge pipeline of great ideas that have been demonstrated in the laboratory to be very hopeful, and now we have to select the best of those and continue to bring them into clinical trials. I can tell you that at Hopkins we have over 15 brain tumor open clinical trials going on right now, whereas we used to have 1 or 2. I think internationally there are many, many very exciting, very interesting, and very new clinical trials being done in adults and children that, if they're successful, will be very hopeful. But the only way we'll find that success is if we push forward and enter patients into clinical trials and ask important questions. But we have reason to be optimistic based on the track record to date.